Yeah, I welcome you all in the week 5th of Bioinformatics for School Children program. Today, for Data Science and Application program, we have with us Jayaraman sir and Sundar sir. Jayaraman sir has 40 years of research experience. Sir started working at National Chemical Laboratory, Pune, in various capacities from 1978 and retired as Deputy Director in 2009. After that, Sir has worked as a CSIR Emeritus scientist and consultant at CDAC Pune between 2009 and 2015. Currently, Sir is working as a visiting professor at Shiv Nada University and as an conjunct professor at Pune University since 2016. Sir's research interest includes chemoinformatics and machine learning, chemical reaction engineering and catalysis, data science and engineering. We also have with us Sundar sir, who has his PhD in computing from National University of Singapore. And since then, Sir worked as research data scientist at various positions at Singapore. And currently, Sir is working as a research scientist at Singapore Management University. We also have with us Kiran sir with us. And with this brief introduction, sir, please can you start today's class?
Okay, thanks a lot. Today we're going to give you a very basic introduction to machine learning in bioinformatics. And machine learning is can be very roughly defined as the ability of the computers to learn from data and identify patterns which are hidden by complexity. Yeah. So it derives its strengths from various uh, faculties like computer science, information theory, statistics, physics, neurosciences, etc., etc. Now, given a set of ECG signals as input, the machine learning algorithm learns from the data and then builds a model, and then the built model can be used to predict the uh, whether a given signal is going to be from a normal person or having a person with a heart disease. So this is one sort of machine learning algorithms which is able to predict from given data, given prior information about the signals labels. I'll come to this a now, little while. Now there are different types of machine learning. One is known as supervised machine learning, for which we know we must have prior information about what we are going to predict. And in unsupervised learning, we need not have information about anything at all excepting the input domain information. And reinforcement learning, which learns from experience, which is different from supervised learning, which learns from data. Now, in today's lecture, I'm going to talk only about supervised machine learning. Supervised machine learning is useful in bioinformatics for various tasks. You can identify protein functions, whether a food protein can cause allergens. You can identify the food proteins which can cause allergen. You can identify peptides which have antimicrobial activities. You can detect different diseases, including various types of cancer. From signals, you can detect heart disease and various types of diseases. You can drug discovery. You can find out the molecules which are active against a particular disease. You can identify protein targets. You can, in neuroimaging, it is useful to identify different types of diseases. You can predict gene functions like whether a given set of sequences are going to be essential genes or non-essential genes, whether a gene is getting localized in which particular tissue. All these things we can identify. Yeah. So we'll talk about supervised learning. Before coming to a bioinformatics example, we'll give you a simple example. One thing is sure, in supervised learning, we require prior knowledge. We'll talk about what is prior knowledge, we'll see. Example, army can reject or select a person based on their height and weight information. Although there are various other attributes they would like, I will explain the methodology only with these two attributes. So, how they have the prior knowledge? They have the historical database of people whose height and weights are known already, and they also know whether performance was satisfactory or not satisfactory. Meaning, given a set of heights and weights from their experience, they know whether a person with this particular height and weight is fit or not fit. So they have a historical database. So here, the domain information. So for every problem, we have to extract domain information. The problem here is whether an whether a person is fit for army or not. That's a question. We are solving that problem. Where identification of a person fit enough for a army or not. This is a problem. Now, the input domain information is the height and weight. And in supervised learning, for a set of people, we have defined about the heights and weights of 6 people. And from the historical database for each of these people, we know whether that the person is fit or unfit. So this is supervised learning. That means for a certain set of examples, we know a person's height and weight. We also know with this combinations of heights and weights whether a person is fit or not fit. Now, with this prior knowledge, with this data, which is now normally known as training data. So, training data in supervised learning means we know the input attributes, height and weight, and output labels, whether a person is fit or not. Knowing this information, we are going to build a model now. Let us see the simplest possible model. The simplest possible model is going to be, I'll come to this first. Yes, the simplest possible model now is what we will see. So let us have a set of people whose heights and weights are known, and then we draw a graph. Now, the labels green and the brown are that of people who are fit or not fit. Now, to Nitya, the question is now, unless why I have labeled them with colors, I'm just drawing. So I'm drawing. I'm just fitting a graph with the data, and I am labeling. Now I can label with green color or brown color because I already know the green color people are fit and the brown color people are not fit. So in only in supervised learning you can color code this. That means you know both height and weight. We also know whether the people are fit or not. So from the data, I have just drawn a graph. Now, Nitya, the simple algorithm here is to and find a surface to separate the data into two different classes. The class of persons who are fit for the army, the class of persons who are not fit for the army. Now, this is now the learning. The algorithm here is a simple line now. So, what is algorithm? You must know the coordinates of the or the equation for the line. Only a particular line will separate the data into two different classes. If you draw if you draw different lines like this, it will not do. So, for example, if I draw a line like this, it will not separate the data into two different classes. So we must know the equation for the line which fits the data into two different classes. Once we know the equation, we can draw the line, and the blue colored examples are new examples. So what we have now done is we have built a supervised learning model with the set of examples are persons whose height and weights are known, and we know certain people who are fit for army, green colored people. We know certain people who are not fit for army, the brown colored people. So with that data, the data now very nicely separates into two different classes because one class of people are getting localized in one area and another class of people are getting localized into another area. So that means we can separate the data into two different classes by just drawing a line. So the algorithm here is to find out which line can separate the data into two different classes. Once you know the line, now you when you get a person for the army, now this algorithm can be put in the computer, and then if you just feed the values of height and weight, you can identify whether the new person is fit for army or not. For example, this blue colored person with this weight and this height, we know is not fit for army, and this blue colored person with this height and weight is fit for army. So supervised learning algorithm, in essence, builds a model with domain information about the problem we are handling, about the inputs. The inputs are height and weight, and the output is decision. So we know both the inputs and outputs. So we are just trying to get a mapping between the inputs and the output. The mapping can be done in several ways and several algorithms. Some problems you can use a linear line to separate the data into two different classes. So in this case, the equation of the line is what we are learning. And then once you learn the equation of the line, you can find out for new examples where we just put it, if the person lies below the line, then he is not fit. Above the line, he is fit. So we build the data with the training data, which we know by prior knowledge or from historical database, and use that data to build an algorithm. Once we build an algorithm, we can use for new examples or new people who are joining army. Just by using a computer program, you can find out whether a person is fit for army or not. Now, let us take examples from bioinformatics. Yeah. So, let us see, so we can be given several sets of attributes for a problem. The problem here is find out whether a person is having diabetes or not. So, this many attributes are normally given, but for the sake of explaining, let us take only two attributes: the blood sugar level and his BP, and the person's BP. Now, you just see here also, we know the input attributes. For all the people, we know the blood glucose, we also know the BP. And we also know whether the person with the glucose level of 148 and BP of 72 is having diabetes. On the other hand, the person with 85 units of glucose and 66 units of blood pressure is not having diabetes. Like that, for all the people, we know that the domain information here is the blood glucose and the blood pressure. We know for all the people with the domain information, and we also know whether these people with this set of values of glucose and the BP is having this thing or not. This comes from prior knowledge that the doctor has or the hospital has of the patients or the normal people. They already have checked. So here we need to have a record of the data. That is the prior knowledge. From the record of data, we know the blood glucose and all the BP. We also know whether a person is having diabetes or not. With this data, let us again try to build a model now. So the red colored people are having BP and the blue colored people are not having BP. So we need to draw a line. Now we have to find out the equation of the line which fits the data into two different groups. Yeah. If you draw lines like this, now it will not separate the data nicely, because here you see, here one person, it predicts this person is not having BP, whereas all these persons are having BP. So which is not correct. Even this line is making mistakes. So we have to draw a line which separates the data nicely into two different classes. So here again, this is the line. This is the best line. This is what we must learn. The essentially y equal to mx plus c here for two attributes. That y is BP, x is glucose, y equal to mx plus c. We must know the slope and intercept of this line. Once you know this, then the line's equation is known. And after knowing the equation of the line, we can identify whether a person is having BP or not by just using a new test example of a person and input his values of the glucose and blood PP and see whether it is lying on above the line or below the line. Once that is known, you can identify whether a person is having a BP or not. Here again, Nitya, are you able to understand what I am saying?
Yes, sir. Yes, sir.
Now, I have one question. Now, how can I color code this red and blue?
Sir, because we priorly had the data that the people with like the red colored dots are a symbol for the people who do have BP, while the blue colored
Very
dots
good.
are the
Yeah,
symbol.
yes, that's what. This is the supervised learning. From the historical database or from the knowledge base of the adapter, we already know certain persons with these values of glucose and BP, whether they have diabetes or not. And what is the prediction for this yellow person?
They have they don't have any
They
BP,
don't
or
have this prediction for this yellow person,
they have an issue with their
and
BP.
he has okay only one is there. Okay. Now, what is his blood glucose units and then BP?
Sir, I'm not sure about that.
This is the blood glucose
Sir, but
50.
we haven't created a line to distinguish between.
What is
Uh,
that? What? Please tell me. I don't understand. Yeah.
So, like, we've created a line to distinguish between the different levels of BP someone has, but we haven't created any line to distinguish whether the levels of glucose in that person is.
See from for the new person, we must make an experiment and find out the levels. Okay. That's why I asked you the question. Unless you have the information of a person's glucose or BP, you cannot predict whether he has diabetes or not. Is that clear?
Yes, sir.
Yeah. Now tell me for these persons before identifying whether he has a glucose, whether he has a diabetes or not, we must know whether his glucose levels and BP by experiments. Okay, by experiments, or that is from diagnosis. Okay, right. So you must measure. You must measure BP using the BP equipment as well as find out the glucose, blood glucose by from from his blood glucose levels. Okay, from a laboratory. From in a laboratory, you have to find that out of a new person. Once you have the information, then you can find out using this information. Just put that point there. That point has the coordinates 50. His glucose is 50 and BP is around 112 or 13. Is that clear now? Nitya?
Yes, sir. Yes, sir.
What was your doubt? Your doubt was how do we know this? Without knowing this, we cannot put it see, that's what I said. No, for we must know this glucose as well as BP. Unless we know that, you cannot predict whether he has got a diabetes or not. For a new person, first just get the measurement and then put it in this graph where he lies. You see, using the coordinates, you put the if he lies here, then he doesn't be this thing diabetes. If he lies here, he has diabetes. Is that clear?
Yes, sir.
Right. Okay. Now, now as I already talked about it. Yeah. Now, with glucose and BP, you could draw a line. Now, this is a line. And if we have, suppose we have three attributes, suppose we are adding, see, more the attributes, the better will be the result. I'll talk about it in a little. Suppose we add skin thickness also. So we have got three attributes. Now you cannot use a line now. So you have to use a plane. So you have to find, now the problem is to identify the plane which separates the data. So now there are three attributes. So one with the three attributes, you can put it in the three-dimensional space. So the red people and the blue people you can separate by using a plane. Here the algorithm is to identify the equations of the plane. Once you have identified the equations of the plane, you can find out for a new person if he lies this side of the plane, he doesn't have BP. I'm sorry, diabetes. If he lies in this side of the plane, he has diabetes. More than three attributes we cannot visualize. But we can always get the equations of what we call it as a hyperplane. We can get an equation for as many number of attributes as possible, like y equal to mx plus c, the equation for two-dimensional this thing for data, for three-dimensional data, we got w1 x1 plus w2 x2 plus w3 x3. Like that, if you have n-dimensional data, you have an equation. Once you have an equation, then you can find out from that equation, and you can, whether using that equation as the algorithm, substituting the values of the attributes like pregnancies, glucose, blood pressure, etc., you can identify the, if the, so we have to find out the equation on the left hand side with attributes, and right hand side is equal to 0. If you put that in that equation, if the value is above 0, you predict this is having diabetes. If it is less than 0, you predict it doesn't have diabetes. So this is a little more difficult. This thing. The idea here is, if you got more than 3 attributes, you cannot visualize, but that exists an equation for a hyperplane. Like an equation exists for a three-dimensional plane, an equation exists for the hyperplane. Using that hyperplane, you can separate the data into two different groups. Right. Okay. Now let us go a little more in detail. Now, suppose you are given a set of sequences, DNA sequences, and you want to identify whether, given a set of sequences, whether they're going to be essential genes. So we have a set of sequences, gene coding sequences like this. This is one sequence with the specific combinations of the ATGCs, right? We have set of sequences, and in supervised learning, so Nitya, I will ask you a question. So we are given a set of sequences. We have to identify whether they are essential genes or non-essential genes. Some genes have essential functions. Some genes do not have essential functions. The genes which have essential functions are known as essential genes. The genes which do not have essential functions are non-essential genes. So the problem here is to identify, given a set of gene sequences, whether that is going to be essential gene or non-essential gene. So, what information you need for a supervised learning, Nitya?
Yes, sir. Um, like like you mentioned earlier, we would have to create some sort of graph and to find out a way to differentiate between the two types of genes.
No, more than a graph, we have to have some domain information. How are you going to extract? I will tell you. We have to have a domain information about a DNA sequence, right? Yeah,
Yes, sir.
that is one. And second, what else you must know for supervised learning? The task is, we are going to identify whether a given gene sequence is going to be essential gene or non-essential gene. So I am going to use a supervised learning. What else I need?
Sir, we'll need some information about basic information about the gene to be able to
Basic
differentiate
information
between
about
the. Sir,
basic information about.
some basic information about the gene because we need to have some data in the first place to be able to differentiate
No,
between
no.
them.
I have a set of genes. What else I need? I will extract. I will tell you how do you extract information. I will tell you. I have a set of genes. I know how to extract information. What else I need for supervised learning? The problem is to identify whether a set of genes is essential or not essential. Yeah. Before that, I'll explain again. Now, for all these examples, I already know the people's height and weight. I also know whether he is fit for army or not. Why I am still building an algorithm? You tell me. Answer the question.
So that even if we introduce a new person, if we enter
Yes. When a
another
new
person
person
in
is
the.
coming, you can find out whether he is fit or not. That is why we are doing it. Understood it here. The same question now I am asking. In genes, I have a set of genes. Okay. I want to build an algorithm to separate the genes into essential genes and non essential genes. So from this, I will extract some information which can be put in, as you say, in a graph. I will do that. What else I need now?
So we can now learn about other genes by putting it.
I need, so I think, like what, apart from height and weight, what did you need in that problem? A person's height and weight is given. What else you needed?
So we needed to know if that person was fit or unfit.
Now here, you are given a set of genes. What else you must know about the set of genes for the problem
So,
here?
so we must know if they're essential or not.
That's what I'm trying to ask you. I want that information from you. So, given a set of genes to build a supervisory learning algorithms, you also must know whether a given gene is essential or not. Is that clear?
Yes, sir. Got it.
Yes. From that we'll start. Now, what we do is now now we have got set of, see, it's a specific combinations of ATGCs. You can extract information which is much easier for you to put it into a computer. Now, one way of doing it is finding out the number of adenines in this sequence. What is the number of adenines? 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Total length is 20. So 6 by 20 is the frequency. Okay, I'm just normalizing it. Similarly, find out the frequency of occurrence of all T's, G's, and guanines, and then cytosins. Okay, is this clear?
Yes, sir.
Yeah. Once you have this, I can have, and see, like this, you can get for other things the frequencies. Once you get the frequencies, now you can solve the problems of essentials and non-essential genes. Okay. The genes have functions like essential genes have functions like housekeeping, etc. They are, some of them are not essential genes. So identification of essential gene is a very important task. So let us give you a simple example. Once you have 10 genes, for all the 10 genes, now I know, this is the question I was asking you. For the first 5 genes, I know they are essential. For the last 5 genes, I don't know, they are not essential. What I have extracted from them, the information about the frequencies of the adenines, cytosins, guanines, and then tyrosins. Once I have this, I can use this now to build an algorithm. Is that clear, Nitya?
Yes, sir.
Tell me, what did I say? What is the process now? What is the problem I am handling, and what is the process? Yeah.
So in the example that you gave us, we have a bunch of genes, and for half of them, these are we know that if they're essential or not, but for
Yeah.
the other half, we're not aware about of that data. So
Right.
to figure it out, the information that we have on the first set of genes,
Hmm.
where we have been given their DNA sequence, and we
So
know
what
whether
is,
they're essential
yeah?
or
What is
not.
the domain information I'm extracting from the input? Yeah.
We're trying to design an algorithm to figure out that all the other genes that we do have, whether they are essential or not.
No, 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 no. I think I did say I'm not giving the information to the algorithm in this form, sequence form. I'm extracting some information. What is it?
Yes, yes. Uh, so the A, T, and G and C, we know the number of times they occur.
So I am finding out the frequency.
Yes,
So
sir.
the importance here is whether the length is 20 alphabets are there, 100 alphabets are there. You can extract only four frequencies. Is that clear?
Yes, sir.
Because it becomes so. Although the sequence length is different, the ATGCs you can get is only same, only four. You get to convert a sequence of alphabets 20 or 20,000. You get only four set of attributes. Is that clear? Frequencies of A's, C's, G's, and T's. Right.
Yes, sir.
Once you have get this, then you can use this to build an algorithm. Let us show it only with uh, frequency of guanine and frequency of adenine. It comes like this. So you have to find out the best line here. So once you know the frequency of guanine and adenine, you can find out whether it's essential or not essential. Okay.
Yes, sir.
I'm just showing it to you with two attributes. We can also do it four attributes, 20 attributes, or any number of attributes. You can find out the equation of a hyperplane, line is separates data which have two attributes. A plane can separate data with three attributes. Hyperplanes can separate data with multiple attributes for which we do not know, we cannot visualize, but the equation exists. Okay, yeah. Now, the same way I have done for height and weight and glucose, I have just drawn this. The only thing is, I have not used the sequence as it is. I have extracted some information from the sequence. Similarly, you can also extract equation from the protein alphabet. You can find out protein functions because there are 20 different alphabets for protein. You can get 20 different frequencies, right?
Yes, sir.
So from those frequencies, you can build an algorithm to separate the data into identify different functions. Sundarda, I think I've already taken up to 540. This is enough. Now you can take over. Am I right, Rajul? I think I've given her the basics of what is um, supervised machine learning algorithm, and I gave her a simple linear classifier. The more rigorousness can come in if we have more classes later. Nitya, if you have any questions, please ask me.
Sir, I wanted to ask, like, we only talked about the supervised machine learning here,
Yeah.
but then there were these other
Yeah,
two that
well,
you
we will.
also
I think
mentioned.
you just have to get a good grasp of supervised learning. For
Yes, sir.
unsupervised learning is a little more difficult to understand. I thought if we have more lectures, maybe we may have in the future, we will explain you about unsupervised learning
Also,
and other learning. Yeah,
sir, I wanted to ask, so is machine learning similar to AI or is it AI?
machine learning is a subset of AI.
Okay, sir.
Yeah. Okay, I'll stop sharing if you don't have any further questions on the slides. Yeah. Mm.
Hi, Prash.
Yes, Prash. Good morning. Good evening. Hello.
Yeah, good evening, sir. Sorry.
Yeah. When were you,
Yeah, I've been.
when did you come?
Yeah, I just arrived, sir. I'm sorry. I've had a very busy day. Yeah, I was
Okay,
just
right.
listening in and out. Yeah, I'm
Okay,
sorry
no
for
problem.
this.
I
Yeah.
have just completed my portion because Sundararajan also has to teach and then show some demonstrations. Thank you. Yeah.
Right. Yeah. Thank you, sir. Yeah, sure.
Yeah, Sundar sir, I'll please request you to take the session now.
Yeah.
Please take the session forward. Thank you.
Uh, Nitya, so we are going for a database project, right? So we are putting some small info on what are the things we are looking for as a database. So we will run through very basic things. Maybe probably you may be knowing, but we just run through it. So any idea on databases, Nitya?
Uh,
What is
Sir,
the data or database or anything? You said you are going through a couple of databases, right? So you went search for online database portals, and then like NCBI, PDB, structure, so many databases are there. So you go and look for a keyword and download the set of data, right?
yes, sir.
So have you ever thought how who is creating all this? snapshot how we go for it. So, as you have seen the biological databases, which are like libraries from information, and then which are basically collected experimental data from various labs and researchers who are working on various disciplines. Like, as you said, you are interested in microbiology or things like that, right? So, everyone is working to their, and then they create some data, they find out, and then they put what they do, they publish in some journal. They want to publish data, what they are finding, and also they get data through experimental technology like high throughput, which will give like genetic information of a particular genome, because genome can we can derive a genome sequence in one go through a lot of experiments. And computational analysis give, we know some part of the data, is it possible to get the other part of the data, something like that. Things are going on where we do build the data through computers, not rather than through lab or anything. So database can be anything. So we get data from various sources, and people are keep updating it regularly, and people also refine it and clean it. That is the more important, because a lot of data which may be not very clean. So we are slightly worried whether the data we are downloading is good enough to proceed our experiments or analytics or something on it. So that challenge is always there in database. So what any database contains? So if you take a database, it can contain like a genomics database can give details about a gene function and things. If you go for proteomics, it will give a structure. What is the structure of the PDP, structure of a protein and things like that. And then there are other databases like metabolics, microarray, phylogenetic, that gives the how the pathway goes on and how the evolutionary pathway is happening from, like we know, like human came from chimpanzee and things like that. So all these are we can analyze the data, and then we can go for localization or clinical. Also we have mutations, like the COVID data is having more mutations and coming out with new types, right? So similarly, other structures and things. So all this data from gives detailed information for a detailed purpose. So we have, if we categorize the databases, we can say meta database, which is a complete information of everything what they are getting, or the based databases, or nucleic acid is based on the ATC, only the nucleic portion of the database, or amino acid goes for the protein details, and signal transaction also there, which gives pathway. And if we categorize the types of database, which give various types of data, which we are able to get through various set of data. Each one has several databases in this format, and we have wiki style or specialized database. So what is the specialized database? That's what we are looking. The specialized database is we narrow down a particular information. For example, you take COVID. Okay, COVID has a lot of information. That may be a picture form or a mutation or structure, or we can have a genetic sequence changes. So many, it can go for from all these databases, types of databases and sources. So all that if I bring in and build a specialized databases, that will be only for COVID. Anything you got name COVID, you will get everything, or whatever you name, like pathway or published literature, everything. So then it's called a specialized database that we are going to build ourselves and publish it. So anyone who want to have a first information on COVID, they can go there and go to our database and get all the information. Also, if someone finds a new sequence, right? So they want to check whether it is a part of COVID published sequence or not. So they can come to our database and match it with a published database of various types of COVID, and then check whether this particular new sequence is also a new type of COVID, or it is similar to the same thing. Maybe 90% it is similar. They may take some mutation might have happened. That's how people have brought out new types of COVID. So our specialized database is the one we narrow down to a particular uh, but all level information, and we categorize and build it in a platform so that people can access. That's our idea. So that's where we want to build our own database on a specific aspect like COVID. We have developed, like earlier batches, they developed Nipah virus also. And last batch, Turner syndrome database. So all these databases are almost ready, and we are waiting for the final analytics tools to it. Then we are going to publish them. We have developed 3 to 5 databases, and 1 or 2 databases we are going to involve you also if you are interested. Then we can move on in the project. Okay. So this is what we call is sub database, means the biological database but sub database which bring a specific with all the information from various places. That's what I just mentioned. Like COVID, we get all the details, and then we add more information to it. Okay, the main thing we want to tell is before creating a biological new database, do not jump into it. Say, hey, somebody has done a COVID database. Okay, I also let me start something. Because the problem is the person would have published it by the time we are developing it. So it is going to be a waste, right? It's no use in recreating a thing which is already on pipeline or somebody is publishing, because they have already benchmarked it, they've analyzed all the databases, and they've done enough study to do it. So it is better not we jump on it before we check overall doing it, any publication has come on it, something like that. So it is better to not to recreate a DB from scratch without knowing what is happening outside. Data quality, because what data we are going to build on, we need to have a good quality of data. That is more important. And know your audience, for whom I am developing the database. Is it for researcher or for a general public? They just want, they don't want any scientific details that they cannot understand, right? So they need to know only what is happening in COVID, whether it will kill a person or it will not kill a person. So you must know the audience and go with modern technology. Means the more refined
Sir, I just wanted to ask, like, when we are creating databases, we're just putting in data onto, like, data we already know, right?
Yeah, data is like, for example, I'm going to show the Nipah virus. So Nipah virus has 9 sequences published. Okay, experimentally validated. So that 9 information we are downloading and building a small database, a sub database, and put it on a web server or publicly, where anyone can access this. So this is the Hey, can you see this? Information, and they combined it and put it in public resource. Mean anyone can access this. That's the idea. So it will go like so it gives like a taxonomy, keyword, citation, and all the information are broken into different category, and then um, so anyone can access the database. Now maybe it may not be working because we are still working on. So it will give all the details. So it has some. We are sorting out the thing, like all the catalog we built on the like keyword. So keyword will have all the keywords available with the, so you can search on based on keyword. So so these are the tools, small tools which we have given that helps to go through all this. See, this will give authors, which are the journals it is published. So from the data, we wrote some Python script to break it, and through SQL we upload to the SQL server in the backend to the database in the server, and then we can have all this kind of, see what are the years and things like that, when it is published, what are the title. So all these are taken from the publication, and we built a data model where we are able to analyze it whichever way we want. Maybe somebody want to say, hey, I want to see Abu Bakr, you made a 13 publication, I want to see. So um, then our database will give all the details. But now it's not working because some connection issues are there with the server, which we are sorting out. Then you can see all the details will come from the database which we launched in the server. So it is clear that this is a small database which we took all the information from other published, and then we refined them. We, even the little girl also went through 3 protein information, and she found whether it is experimentally validated. And then we added to the we have 9 protein sequences in there. So this is the sub database which I am talking. Like we are going to have a project on plant. If you are interested, plant database which we published 10 years back. Um, Now we want to revamp it. Means make it new and add some more tools and republish it. So if you want, you can work on it, or you can join the other Naya Mehta last time student. She started the Turner syndrome database. That still we need more data to be added to the database. Then we can publish it. So these are the things we are looking now. Yeah. So the idea is to pinpoint what we want to do. As I mentioned, we need to check whether anybody has already published it better than us. Then we don't waste our time. We just go through the database. And if you want to make something give more value to the researcher who are coming to our database, that gives a quality and value added, so that our work can be published in a better journal and things like that. So we were talking about lot on data. So can you tell me which are these are data, which can be called as data? Number or text or paragraph, image, audio, video, stationery, ranking, school. Can we? Which are the things we can call on the left as data?
Sir, I think all of them in a way are data that we can utilize.
Yeah, accurately. Yeah. So anything you name these days, anything you can add it as a data. Means even image nowadays, like Facebook or anything, they handle a lot of images. That is also a data. And video also. We can search a lot of MP3 and MP4 or floating around. We can have everything is data. Anything you want to search, you want to go through, is already there. So everything like Facebook or Twitter, Instagram, they all keep improving their quality so that the customers can get into what they want to go through or do something like that. Okay. So when we call after data, we call database. Before going to database, we call table. So on the right side, we have a table that gives class and mathematics, science marks. So we have 5 students who got marks in different subjects. So what it tells, so we once we get data, then we collect according to a row and column. Means column gives what are the things like information on maths or science or address of a, something like that. And who is for whom? It is for a student 1 or 2 or so this way we arrange it. Why we arrange? Later only we can find if you want to find the student 1 number, immediately we can pull from the database because it is only 5. But if you take a school, you can see how many classes into how many sections, how many standard. It will grow a very big database, right? So if you want to know who are the rank holders for each class or each standard, like grade 5 or 6 or 7, like that. So once we have the data like this, then it is pulling information and make a graph or anything. It becomes very easy, right? So that's what we want to do. So we built a small school database where we insert all the information, whatever is available per semester, per test marks and everything. So it gets accumulated like this. Table and each table has a row like student 1, 2, 3, and the column gives various information. It can be a subject marks or a location or where the address or parents number, everything. So in general, data is a plural of data that comes from facts and statistics. I mean, we have the fact and statistic is a number or a value based. What that represent for a particular student or anything. It's a fact. It's a real derived numbers. So if we go in a tabular way, we say table that contains header, rows is for each student, and column is, if we say protein, so protein 1, protein 2 also can have, and we can have thousands of column to give various information. So we put it in this format because that is important later to analyze, right? So it should be a clean thing. That's what I say. Student 1 detail should not be mixed with student 2. Then we cannot derive anything, right? So that and for pertaining to a student 1, we have a and for pertaining to a student 1, all the details should be correctly derived and then updated into our database. That will give even like a Excel. It is representation is like a Excel. You write a column and row and you keep adding things, right? Then you derive some numbers or graphs or same thing database. Database means the advantage is it's queryable. Means we can give a query or analyze it or give it some analytics. That's the advantage of database. So next step is database. So database may have multiple tables. Say for student marks, we have a table, and student information, we have another table that gives only address of a student and a contact number, parent contact number. If anything happened to the student or on emergency or something, or the school wants to talk to the parent, so they keep an important contact number for each student in a different table. Because why we need different table? Because table one, student marks can repeat over many things like term exam or semester, all this. But student detail is fixed. It is fixed where he stays and whom to contact. So this gives a unique information for each student. So that's why we keep the table separately. Uh, any question?
No, sir.
Just 1 second. I give a 1 minute break. Just 1 minute. Just 1 minute. I give you a 2 minutes break, then we'll continue. Okay.
Okay, sir.
Yeah, yes. In the meantime, I think we all should give a thumping round of applause to. Sorry, what is this? Why is it? I need to change it. Yeah. Yeah. My face is clear. Yeah. What I was saying is, and I think we all should give a thumping round of applause to Nitya, who has come on time. Rajul, what do you think? Yes, I think. Yeah, I think we should give real hands. Nitya, what do you think? I think we all will clap you so that it will hear wherever you are. Okay? Yeah.
Thank you, sir.
Yes. And also three cheers to Jayaraman sir and Sundarda as well.
Yes.
Yeah.
Maybe we can take a picture, photo.
Sure, sir.
Yeah, sure though. It's
Uh,
always somewhere in the beach and he doesn't take us.
yeah, beach is a good place.
It's a pity that you only are enjoying there. You don't take us there.
Yeah, if there are too many people, then enjoyment is gone. Okay, Kiran and Nitya, you can switch on the camera so that Rajul will take
Yeah,
a
I'll
snap.
take.
I'm trying. Okay. In a minute. Okay. Hello.
Yeah, hi Kiran. Yeah.
Hi.
I took the picture. Thank you, sir. Thank you so
Thank you,
much.
Rajul. Yeah.
So, is now the session is over or? Right.
A few more minutes.
Okay. So at some point, if Nitya still have time, I want to talk 5 minutes because the project and also the next thing also we want to discuss. So if she, okay, I'll wait after Sundar does.
Okay, Nitya, you understood this, right? The table concept.
Yes, sir.
So we have multiple tables because we need to keep information because we don't do repetition. We are very strict without wasting a lot of space. If we merge the table 2 with the table 1, then the address will be repeated so many times, right? So that we don't do. And we do a merge of database tables when we need it. Okay. So the UniProt is the one where a lot of protein structure details are there, and we all our database usually we use the UniProt because UniProt has a specific easy to handle text format. And the first two column gives what it is, whether it is a author name or a keyword name, all these So it gives the index abbreviation. All the lines are with that. But other like next one is the NCBI is more having a lot of database information, but it's not very clean or unfriendly. I mean, the structure is very difficult to handle by Python code or anything. But still we can do, but too much complicated. And also many of the NCBI data we found which are repetitive and also not well defined or clinically or experimentally validated, because any author they want to get something, they just want to push it as with a number into NCBI before they complete their experimental with other labs or anything. So that's why I found NCBI has more data which are not very clean or clear. So rather I go for the this one Uniprot, which has a specific, because they do a background search before adding to the Uniprot database. So we get a more better and clear data rather than everything available. So what usually I suggest students to go for the Uniprot first. Later, if we find anything good, we can add from NCBA. For one particular project, for the hypothetical protein, we are trying to do, because we got about 1,000 Uniprot data and another 600 from NCBI. Still, our researchers are sorting out to add them together to make a new combined database. So it is taking more time now. Maybe you all children can help because you have more time. If you can add more students, then we can speed up things. Okay, any question on that? Develop our specific particular data base and put it in the web server. As I shown, like a NIPAM, we get sequence data, we get author data, we get publication detail, and everything, whatever we talk about that NIPA, we get into one platform and publish publicly so that anyone can go through it. Okay, any question?
No, sir, I got it.
So, Rajul, what is the timeline? 1 hour total, is it? Okay, then I don't go in much advanced thing. Nitya, you can decide which project you are going to do, as you are going through a lot of modules, right?
Yes, sir.
So you can decide which subject, which teacher you want to continue. You can, if you have time, you can contribute for the database also, which we are planning some of the databases, which will be giving more information. Because these databases or anything, right, work you can submit in our in BIXS'23, going to be in VIT Vellore during November 24 to 26, right, Rajal? Yeah. And then you can physically go there and present it also, or you can do a hybrid, like you can from your home you can present it in the conference. Because during corona and all, we got children to we allowed children where the Nobel laureate were quite impressed and see how a 10-year-old can present a paper in the conference. He got amazed actually, and that's how we train kids and students and make them a buddy researcher, so that also you can do a contribution. As you have seen, we have done 3 databases already fully, and we are going to publish them. I'm trying to add some tools which is having some server issue. Then it should be sorted out. Okay.
Yes,
Any
sir.
final comments from Prash? Prash, are you there?
Yeah, no comments so much, all well. And I think, so what I was just wondering is, Rajul, how about other 4 students? Would we be sharing the recording and their hands-on, how is it to be done?
Yeah, I'll be sharing the recording.
Okay, yeah. And then Jayaraman sir, Sundararajan and Kiran will probably help them, or are we going to have any special class if they want back, or I don't know.
Yeah, sir. Once yes, so once we have sessions more, I'll ask the students pick if they want to pick their favorite subject, or else we'll just do as designated.
Next will be me. I can spare some time for them as well. I mean maybe Sundar can, maybe Jayaraman sir can be there for hands-on or something like that. I'll take about 1 hour 15 minutes next Saturday. Yeah.
Yes, sir.
Yeah.
Kiran sir, would you want to share
So,
something?
yeah, what I was saying is
Yes.
next Saturday is my talk, right?
Yes, sir. Yes, sir. Sorry.
So no, since I thought since I'm the only one who will be giving the talk, so I can spare 15 minutes for them towards Jayaraman sir's hands-on if they want. Okay.
Okay,
Yeah.
sir. Thank
So
you, sir.
5 to 6:15, I'll wrap it up. Okay, and then we'll see. Huh? Yeah. Thank you.
Thank you, sir.
Thank you so much, Sundara and Jayaraman sir, for the wonderful talks. Yeah.
Thank you, Nitya. Thank you, sir, and Kiran.
Okay.
Rajul also for all the help. She's ever-ending help, never tiring helper.
Thank you, sir.
Thank you, Nitya. I'll be
Thank you,
putting the
sir.
information. Rajul Di will put all things in the Google, the group also, right? Google,
Okay, sir.
the Google group we have, so that anyone can go through later on they want to go through all the syllabus, and I will send the PowerPoint also. Later she will put it there. And you can think, Nitya, whether you want to join which project. Slowly you can start now. We have 3 more months to. You can have a good project done, and then we can present. You can present it in the international conference. Okay. Okay.
Yes,
Thank
sir.
you, Nitya. Thank you.
Thank you so much, sir.